All right, let's talk about something that is both a medical miracle and, honestly, a potential minefield: blood transfusions. They save lives every day, but what happens when this incredible gift goes wrong?
Get this. Every single year, just in the US, we're talking about over 15 million pints of blood being transfused. I mean, that's a staggering number. It's a total cornerstone of modern medicine, a procedure that's happening constantly, from the OR to the ER, saving lives. But what happens when the body fights back, when this life-saving gift is rejected? The immune system can launch this fierce counterattack. And let me tell you, when it comes to transfusion reactions, timing is absolutely everything. So think of this timeline as our roadmap for today. We can basically classify every transfusion reaction by when it shows up. Are we talking minutes, the first few hours, or something that kind of sneaks up on you days or even weeks later? This clock right here, that's going to be our guide. Okay, so first up, the immediate dangers. These are the ones that hit fast. I mean, scarily fast. We're talking about reactions that can happen before that bag of blood is even empty. And the fastest one of all? That's the anaphylactic reaction. Basically, it happens when a patient is missing a specific protein, something called IgA, and they get blood that has it. Their body just freaks out. It sees it as a massive threat, and boom, you get shock, crazy swelling, trouble breathing, all within seconds. I love this quote. It just perfectly sets the stage for our next immediate danger, the acute hemolytic transfusion reaction, or AHTR. It is a truly devastating mistake made by the immune system. So what's going on here? Well, it's a type 2 hypersensitivity reaction. And you know what the worst part is? It's almost always because of a simple clerical error. Someone gets the wrong blood type. The patient's antibodies just immediately go to war with the donated red blood cells, causing them to literally explode. This releases all this toxic hemoglobin that wrecks the kidneys, and it can kick off this horrible clotting cascade called DIC. It's a total disaster. All right, so let's move along our timeline. We're now in that first 6-hour window after a transfusion starts. This is a really interesting period because it's where we see the most common reactions, but also some of the trickiest ones to figure out. So, the two you'll see most often are 1, a febrile non-hemolytic reaction, which is basically just a fever caused by inflammatory proteins called cytokines, and 2, an urticarial or allergic reaction, hives, itching. The good news? Both of these are usually pretty straightforward to manage. But in that same 6-hour window, something much more serious can happen. Picture this. A patient is getting blood, and all of a sudden they just can't breathe. You get an X-ray, and you see it. Their lungs are filling up with fluid. This puts the medical team in a really tough spot. They have to answer a critical question and fast, because the answer could be life or death. Is this an immune attack on the lungs, what we call TRALI, or is it more of a plumbing problem, just too much fluid, what we call TACO? So how in the world do they tell them apart? Well, think of TRALI as the immune attack. Donor antibodies are attacking the lungs, making them leaky, and that causes low blood pressure. Now, TACO is all about volume, too much fluid, too fast, and that causes high blood pressure, and you can actually see the neck veins bulging. Getting this right is, you guessed it, absolutely crucial. Okay, so we've made it through the first few hours. The immediate danger seems to be over, but the story isn't finished yet. Now we need to look at the complications that can pop up days or even weeks down the road, sometimes long after the patient has already gone home. This next one is kind of like the immune system's long-term memory kicking into gear. Imagine a patient was exposed to some minor thing on a red blood cell in the past. Their body remembers it. So when they get new blood, it mounts this slow, smoldering attack. It's usually a lot milder, and sometimes you don't even see any symptoms at all. And then there's graft versus host disease. Now this is rare, thank goodness, but it is devastating. I love the metaphor of a coup for this one, because that's really what it is. The donor's immune cells, their T lymphocytes, get into the patient's body, and then they start attacking the patient's own body, as if it's the enemy. Okay, so we've talked about all these pretty scary risks. The big question is, how do we fight back? Well, let's open up the playbook. Because when you're talking about transfusion reactions, an ounce of prevention is worth a pound of cure. It really is the best medicine. Hey, remember those common fevers we talked about? The ones caused by cytokines from the donor's white blood cells? Well, the solution is actually pretty simple, but it's super effective.
It's called leukoreduction. We just use a special filter to pull out those right blood cells before the transfusion ever happens. You're stopping the problem right at the source.
And what about those really severe anaphylactic reactions, the ones in patients who are missing that IgA protein? The strategy there is washing, and it's exactly what it sounds like. The red blood cells are literally washed with a saline solution to get rid of all the plasma and those IgA proteins that were causing all the trouble. Simple, but life-saving.
And finally, how do you prevent that cellular coup, that graft versus host disease? You use irradiation. We give the blood a controlled dose of radiation, which basically neutralizes the donor's T lymphocytes. It makes them unable to launch their attack on the patient's body. It's like a targeted strike to keep our most vulnerable patients safe. Okay, so let's quickly recap the big points. First, reactions are all about timing, and the ones that happen right away are usually the scariest. Second, telling the difference between T-Raleigh and TACO is a huge diagnostic challenge for doctors. And third, and this is the most important part, we have a very specific playbook to prevent these things. Leukoreduction, washing, and irradiation. Each one solves a very specific problem. And that brings us to one last thought. With everything we now understand about these risks, and with this amazing playbook to prevent them, blood transfusions are safer than they've ever been. So it makes you wonder, what's the next frontier? Could we one day create a truly universal blood type? Or maybe even synthetic blood to make this whole life-saving procedure virtually risk-free? Something to think about.